Hello everyone, my name is Fernanda Rosa, and today I will share with you how a fecal microbiota transplantation can impact the intestinal microbiota and blood biomarkers in pre-weaned dairy calves. Fecal microbiota transplantation, or FMT, is a procedure by which microbiota present in the stool from a healthy individual is transferred or inoculated into the gut of a recipient. This FMT procedure has been used to treat inflammation of the colon in humans infected with Clostridium, as well as to treat inflammatory bowel disease. Recently, FMT has been used in clinical trials in infants in order to restore the beneficial microbiota acquired through vaginal birth in C-section delivered babies. Therefore, we aim to evaluate if dairy calves can also benefit from fecal microbiota transplantation, as we have seen in human trials, as well as if FMT in neonatal calves can shift the gut microbial composition. To address those questions, we select a cow from the herd that was a high-producing cow with no clinical history of metabolic or inflammatory diseases, and the fecal samples from that cow tested negative for harmful pathogens. Therefore, that cow was a fecal material donor. We mixed 25 grams of that fecal sample into the antibiotic-free milk replacer offered to the calves from 8 to 12 days of age, and we evaluated the growth and health performance, the immune response, and the microbiota composition. We used a total of 8 animals per group. All the calves were housed in individual calf hutches, and all the calves were weaned at 7 weeks of age. The calves in both groups, control and FMT, they all received the same commercial antibiotic-free milk replacer, with the exception that in the morning feeding, the FMT group from 8 to 12 days of age were also inoculated with the fecal material. Once per week, we collect blood and fecal samples from all the calves for further analysis. Now, starting with our first set of results regarding to the overall growth performance of the neonatal dairy calves, let me show you how the plots are presented. In the x-axis, it's the week of age, y-axis, the measurement. In each plot, there is the statistical significance statement. The black arrow indicates when the FMT treatment is started. The orange dots represents the calves that receive FMT, and the dark dots, the control calves. We observe a tendency for a greater body weight and weight high over time in the calves that receive FMT compared with the control calves. Before we move on for the blood biomarker results, I'd like to highlight that during an acute inflammation, for example, within the gastrointestinal tract, the epithelial cells and the resident immune cells, such as macrophages, through their tool-like receptor pattern recognition, they will release pro-inflammatory cytokines, such as interleukin-1 and interleukin-6, that can stimulate the migration of more immune cells from the bloodstream into the site of infection. At the same time, that those cytokines, they can serve as a signal to the liver of the inflammatory condition. And the liver will respond upon that stimulation with an increase in the production of the positive acute phase proteins, such as haptoglobin, while decreasing the production of the negative acute phase proteins, such as albumin. Therefore, we evaluated those inflammatory biomarkers in the blood of the calves. We have previously reported that during the neonatal period, meaning during the first three weeks of life, the calves that receive FMT had greater haptoglobin concentration in the blood when compared with the control calves at 2 weeks of age, while a tendency for a greater concentration in blood of interleukin-1 beta was observed for the FMT calves compared to the control at 2 weeks of age as well. Now, when we look to the same pro-inflammatory cytokine, interleukin-1 beta, and the other cytokine, interleukin-6, during the weaning period at 6 and 7 weeks of age, we observed a lower concentration in blood of those cytokines in the calves that receive FMT compared with the control. Therefore, the take-home message from this first set of results is that during the neonatal period, FMT induced temporal changes regarding to the inflammation, such as the increase in haptoglobin in blood, as well as interleukin-1 beta. However, during the weaning period, interleukin-6 and interleukin-1 they were lower in the blood of the FMT calves. Therefore, these are positive results, with the meaning that FMT improved the response to inflammation relative to the control calves. Now, switching from the blood biomarkers to the gut microbiota composition analysis, here are the plots for the alpha diversity indexes, where on the left, we have the observed operational taxonomic unit measurement, which is the measurement of the different species in each sample, in this case, in each group per week. And on the right, we have the measurement of the diversity richness of the microbiota composition. And for both cases, we observe that prior to FMT, at the week 1 compared to the week 5, the group that received FMT had an increase in the alpha diversity indexes of microbiota. Looking in the overall microbial composition analysis per week in both control and FMT animals, as well as with the donor cow, we observed that the phylus Firmicutes and Bacteroidetes were the most abundant ones in all the calves, which was similar with the fecal microbiota composition of the donor. However, the abundance of this family within the Proteobacteria phyla decreased from the week 1 to the week 5 in the calves that receive the FMT. And this is a positive finding because others reported that Proteobacteria microbes are associated with calf scours. And recently, another study demonstrated that the same family from the Proteobacteria phyla, which we observe in the FMT treated calves, is the key contributing factor when calves have diarrhea. So in summary, FMT in calves induce changes in the gut microbiota profile, as well as has positive effects on the response to inflammation. Therefore, we can conclude that FMT might be used as a therapy for neonatal calves to improve the host performance. With that, I would like to acknowledge South Dakota State University, as well as the Agricultural Experimental Station of SDSU, all the staff of the Dairy Research and Training Facility of SDSU, and Dr. Osorio's laboratory for all the help and funding for this project. And with that, I would like to take any questions that you might have in the following Q&A. Thank you.